Hello, this is Matt Dean with A Plus College Ready, and today we're going to talk about cellular respiration. Cellular respiration is the process of releasing the energy that's contained within organic molecules. We usually think of it as glucose, but it can also be proteins or lipids. This is an example of a catabolic reaction, a reaction that breaks down molecules and releases energy. The process uses the energy from the organic molecules to make ATP. ATP then serves as the energy source for almost all of the body's endergonic or energy requiring reactions. Cellular respiration also releases heat, an unusable form of energy, and free electrons. And these free electrons serve as the source of energy for producing the ATP the body needs. We'll first talk about the aerobic version of cellular respiration. When oxygen is present in the cell, aerobic respiration takes place. The first stage of the process is glycolysis. It occurs in the cytoplasm or the cytosol. And the other two stages, the Krebs cycle or citric acid cycle and the electron transport chain, they take place within the actual mitochondria. We'll talk about some exceptions to this later on. Aerobic cell respiration has the chemical equation given here. So the reactants for aerobic cellular respiration are oxygen gas, glucose, and the reaction will produce the main product of free energy, which is usually captured in the form of ATP. It'll also create the waste gas, carbon dioxide, some water vapor, and some heat energy. This reaction has a delta G or Gibbs free energy value of negative 686 kilocalories per mole. The negative tells us that the free energy is released. It's available to do work. And it's this free energy that's used to make the ATP. Now there's also anaerobic cellular respiration. When oxygen is not present, anaerobic respiration takes place. Now, just like aerobic respiration, the process starts with glycolysis. So glycolysis is the first step of all cell respiration. Every organism that we know of, from bacteria all the way up to human, all carry out glycolysis. And in all organisms, this process is carried out in the cytoplasm or cytosol. The second step of the anaerobic process is called fermentation. It too occurs in the cytoplasm. It's important to note that some books, some questions refer to all of anaerobic respiration as fermentation. So it's really important to pay attention to the context of the question when answering questions about anaerobic respiration. The process of cellular respiration is highly conserved across all forms of life. Almost all organisms, prokaryotic and eukaryotic, do glycolysis in almost exactly the same way using the same enzymes. The enzymes and chemical reactions of the Krebs cycle and the electron transport chain are also highly conserved across most of life. This high level of conservation indicates that all living things are related and that somewhere back in the evolutionary history of life, we all shared a common ancestor which did cellular respiration this way. Now, before we get too deep into cell respiration, we need to talk a little bit about redox reactions or oxidation reduction reactions. During cellular respiration, glucose or our other organic molecules is oxidized. Let me correct there. Oxidation is the process in which electrons and energy are removed from a molecule or atom. Hydrogen is also usually removed. Whenever a molecule or atom is oxidized, another molecule or atom must be reduced because the electrons, the hydrogens, they can't just disappear. The process is called oxidation because oxygen is very good at removing electrons from other atoms or molecules. But it's important to note that not all oxidation reactions actually involve oxygen at all. Reduction is the opposite of oxidation. During reductions, electrons, energy, and also usually hydrogens are added to a molecule or atom. During aerobic cell respiration, the electrons and energy that are removed from the glucose are first used to reduce electron carriers, either NAD+ to make NADH or FAD to make FADH2. Ultimately, the electrons provide the energy to produce ATP. And the electrons, in the end up combining with oxygen gas and hydrogen ions to make water. It's important to note that reduction is called reduction because the addition of electrons to an atom or a molecule decrease its overall charge. They make it more negative. So here we see an organic molecule, think of this as glucose, getting oxidized, going from left to right. Those electrons are then added to NAD plus to make NADH. So as the organic molecule is being oxidized, the electron carrier is being reduced. So a lot of times we'll use the acronym oil rig to help us remember how oxidation reduction reactions work. Oil rig stands for oxidation is the loss of electrons, while reduction is the gain of electrons. So remember oil rig, and that'll help you understand oxidation and reduction. So let's get into the details of aerobic respiration. First of all, we have glycolysis. During glycolysis, the glucose is split, it's oxidized. Glycolysis occurs in the cytoplasm of every cell. Krebs cycle, or the citric acid cycle, during that process, the pyruvate, which we'll talk about a little bit later, that's formed during glycolysis, is oxidized. The electron carriers are reduced, and this process occurs in the innermost part of the mitochondria, or the matrix. Now, in bacteria, this occurs in the cytoplasm as well. The electron transport chain, the final step of aerobic respiration, during this process, electron carriers are oxidized, and the energy released is used to make lots of ATP. This occurs on the cristae of the mitochondria in eukaryotes and on the inner folds of the cell membrane in aerobic bacteria. So let's get into some details about glycolysis. Glycolysis is typically divided up into two phases: the energy investment stage, which we'll talk about first, and later we'll talk about the energy payoff or energy harvesting phase. Let's talk about the energy investment stage first. So we start with glucose. To begin the process, the glucose is first phosphorylated. So phosphate groups from ATP are connected to the glucose. And notice when the first one of those happens, the glucose is now called glucose 6-phosphate. Changes shape a bit, and it's phosphorylated again to become fructose 1,6-biphosphate. So these two ATPs, we might think of them as activation energy. To get the process started, two ATPs have to be used. So that's why this is called the energy investment stage. Now there's an enzyme called phosphofructokinase which carries out this second phosphorylation step. This enzyme has a feedback or an allosteric site on it, which is really important in regulating the rate of cell respiration. We'll revisit phosphofructokinase a little bit later. The next part of glycolysis is called the energy payoff or energy harvesting phase. And let's talk about this diagrammatically. So let's go back to our diagram here for a second. This fructose 1,6-biphosphate changes shape slightly and becomes two molecules. Each one becomes a molecule of glyceraldehyde 3-phosphate, or we usually call this G3P. Remember that in photosynthesis, G3P is sort of like half of a glucose with a lot of energy. So during this first process, the G3P is oxidized. And that means electrons and energy and hydrogens are taken from the G3P and they're transferred to the electron acceptor NAD+, to make the energy storage molecule NADH. Also right in here, some energy is taken directly from 1,3-biphosphoglycerate to make ATP. And I want to go back to the previous slide for a second. That process of making ATP is called substrate level phosphorylation. In substrate level phosphorylation, phosphate groups and energy are transferred directly, in this case from 1,3-biphosphoglycerate and later from another chemical called phosphoenol pyruvate to ADP to make ATP. And we'll talk later about chemiosmosis, another way of making ATP. This is not that. This is transferring energy and phosphate groups directly from one chemical to ADP to make ATP. So we see that happening again right here. So there are three main products of glycolysis. For each glucose that goes through the process, glycolysis produces 2 molecules of NADH. It produces 4 ATP molecules. But remember, in the previous process, we used 2 of those. So a lot of times we'll say per glucose, glycolysis nets 2 ATPs. And what's left of the sugar are 2 molecules of what we call pyruvates, or sometimes they're called pyruvic acids. These are 3 carbon molecules, sort of like half a glucose. When glycolysis is over, most of the energy that started out in the glucose is still within the bonds of these 2 pyruvate molecules. Glycolysis gets out a little bit of the energy, but most of the energy is still contained within the bonds of these 2 pyruvate molecules. We talked a minute ago about electron carriers. The NADH formed during glycolysis is formed by the oxidation of G3P and the transfer of the electrons and energy from to NAD+. So this is NAD+. And we can see that it's being reduced. So it's gaining electrons, it's gaining a hydrogen, and it's gaining energy, and it becomes NADH. Now later, that process will run the other way, and the NADH will be oxidized, and that the energy there will be used to create ATP. We'll talk about that in a minute though. So here we can see some substrate level phosphorylation. So we have an enzyme here that's grabbed on to the phosphorylated substrate. It's also grabbed on to ADP. And in this process, this phosphate is transferred from the phosphorylated substrate to ADP to make ATP. So summarizing glycolysis, when it's over, one glucose molecule yields 2 molecules of pyruvate, 2 molecules of electron carriers or storage molecules, and 2 net molecules of ATP. And remember that at the end of glycolysis, most of the energy that was originally in the glucose is still contained within the bonds of the 2 pyruvate molecules. So let's take a sidebar and talk a minute about mitochondria. We know in eukaryotic cells, the Krebs cycle and the electron transport chain phases happen within the mitochondria. Remember that the mitochondria is an organelle. It's a membrane-bound organelle that has its own DNA, its own bacteria-like ribosomes, and its own enzymes. And it can also reproduce independently of the cell it lives in via a process that's almost identical to binary fission. That's important because we think that mitochondria were originally aerobic bacteria that lived independently, and that somehow a cell took them in, and that over time they became dependent on each other, and that's how eukaryotic cells came to be. That idea is called the endosymbiotic hypothesis. Now, the mitochondria has an inner membrane that's folded into these little folded structures, almost like the villi in the intestine, called cristae. And those folds are about increasing the surface area, because that's where the electron transport chain takes place. The innermost part of the mitochondria, the innermost compartment's the matrix, and this is the location of the Krebs cycle or citric acid cycle. It's also important, though, to remember that there are some bacteria that do aerobic respiration. They don't have mitochondria, so the Krebs cycle in them happens in the cytoplasm. And the electron transport chain actually happens on folds of the cell membrane itself. So here's the mitochondria, and we can see the outer membrane. We can see this folded inner membrane in here with all its surface area. We can see that the mitochondria has its own DNA. It has its own ribosomes, and it's very much like a bacteria. Now, we said a second ago that step 1 of aerobic respiration is glycolysis, and step 2 is the Krebs cycle or the citric acid cycle. But in between those two steps is a short part of the process that's referred to as the pyruvate conversion. You might also hear it called the preparatory step or the transition step. Before Krebs cycle can start, the pyruvate, which is a 3-carbon molecule, has to be converted to a 2-carbon molecule called an acetyl group. This process occurs in the mitochondria. During this process, the pyruvate is oxidized. So 2 electrons from each one of those pyruvates are transferred either to NAD plus to make NADH or to FAD to make FADH2. Each pyruvate then releases one of its carbons as carbon dioxide gas. What's left behind is a 2-carbon group called an acetyl group. That attaches to a coenzyme that's already in the mitochondria to form a molecule of a substance called acetyl coenzyme A. The acetyl coenzyme A molecules serve as the starting points for the Krebs cycle. So even though the pyruvate conversion isn't considered a step, a phase maybe, it's a short process that occurs between glycolysis and the Krebs cycle. It converts the 3-carbon pyruvates to 2-carbon acetyl groups so that the Krebs cycle can start with a 2-carbon acetyl group. This diagram sort of shows that happening. So here's our 3-carbon pyruvate. Here it's getting oxidized. The electrons are transferred from the 3-carbon pyruvate to NADH, to NAD plus to make NADH. And one of the 3 carbons, the one in blue in this case, is released as carbon dioxide to leave behind a 2-carbon acetyl group, which attaches to this coenzyme A to make acetyl-CoA. And we can see that happening here. So here's our pyruvate. It moves into the mitochondria where the pyruvate conversion happens and it forms acetyl coenzyme A. At that point, the cell's ready to carry out the Krebs cycle. So the Krebs cycle, as we said earlier, occurs in the matrix of the mitochondria in eukaryotes. It occurs in the cytoplasm of aerobic bacteria or aerobic prokaryotic cells. During the Krebs cycle, the 2-carbon acetyl coenzyme A molecules are oxidized. Their electrons and energy are transferred to the electron carriers, NAD plus to make NADH, or FAD to make FADH2. 2 molecules of ATP for every glucose are also produced by the process we discussed earlier, substrate level phosphorylation. During this process, phosphate groups are transferred from an intermediate called succinyl-CoA to ADP to make ATP. By the end of Krebs cycle, all of the carbon atoms from the original glucose have been released as carbon dioxide gas. And most of the energy that was originally in the glucose by the end of Krebs cycle is in the form of either NADH or FADH2. Now, if you look at this diagram of the Krebs cycle, it's very complicated. You don't need to know all these steps. What you need to know is the process begins with a 2-carbon acetyl group attaching to a 4-carbon group. You don't need to know it's oxaloacetate, but those form a 6-carbon group called citrate. That's why Krebs cycle is sometimes called the citric acid cycle, because it essentially starts with 6-carbon citrate. At almost every step along the way, you see that the boxes in blue, the sugars or what's left of the sugar, they're oxidized, and that the electron carriers are reduced. So electrons and energy are transferred from the materials in the green boxes to the electron carriers. There's also some energy in here transferred directly from succinyl-CoA to ADP to make ATP. So by the time the Krebs cycle is over, most of the energy has been transferred to these electron carriers and to a little bit of ATP. And what's left of the sugar is released as carbon dioxide gas. So kind of keeping track of the products. For each molecule of glucose that entered glycolysis, Krebs cycle produces 4 molecules of carbon dioxide, 6 molecules of electron carriers in the form of NADH, 2 molecules of FADH2, and 2 molecules of ATP. And again, most of the energy that was originally in the glucose by the end of Krebs cycle is contained in these electron carrier molecules. So at this point, the cell is ready to carry out the electron transport chain. Remember, this occurs on the folds, the cristae of the inner mitochondria in eukaryotes, and on the folds of the cell membrane in aerobic bacteria. During the electron transport chain, the electron carriers, NADH and FADH2, are oxidized, and the energy from these molecules is used to generate ATP. The actual electron transport chain is composed of a group of proteins called the cytochromes. And these proteins are highly conserved in almost all organisms because of common ancestry. We all evolved from a common ancestor. So let's talk about how this process actually happens. The process starts when one of the electron carriers is oxidized. We'll start with NADH. And it's oxidized by the first cytochrome in the chain. This is sometimes called, the cytochrome is called FMN sometimes. The two electrons lost from the NADH pass through three proteins, or proton pumps, as they move down the ETC. The electrons move from the least electronegative location, the first cytochrome, toward the most electronegative location, the last cytochrome. As the electrons move through these proton pumps, their energy is used to power the pumping of hydrogen ions or protons from the matrix of the mitochondria into what's called the intermembrane space, the space between the outer and inner mitochondrial membranes. A single proton is pumped as a pair of electrons travels through each of the three proton pumps. The pumping of these protons creates an area of high proton concentration in the intermembrane space and an area of low proton concentration in the matrix. And this is an ideal way to store potential energy. The bigger the gradient, the more potential energy is stored. Think of this as almost like stretching a rubber band. And the more it gets stretched, the more energy it stores. An enzyme called ATP synthase is embedded in the inner mitochondrial membrane. It has a channel which will allow protons to move in through it. This channel is the only way back into the matrix for the hydrogen ions or the protons, because the inner membrane is mostly nonpolar and it won't allow protons to go through it. So when that channel is open, the protons will move very quickly from the inner membrane space through the channel back into the matrix. Now this enzyme is unique because it's able to use the kinetic energy of these protons or hydrogen ions moving through it to add phosphate groups to ADP to make ATP. The energy from a single proton can provide the energy to create a single ATP. The process of using this energy from the proton or hydrogen ion gradient to make ATP, to phosphorylate ATP, is called chemiosmosis. That's a really important term for you to understand. So the electron carriers are oxidized, protons are pumped into that intermembrane space, that builds up a gradient. Some of the protons shoot back into the matrix through the ATP synthase channel, and the energy from those protons is used to add phosphate groups to ADP to make ATP. This process is the most important mechanism for making ATP. Almost all of the ATP made during aerobic respiration is made during this step of the process. This process of chemiosmosis is also how ATP is created during photosynthesis. So a couple of other details. Each NADH ultimately provides the energy to create three molecules of ATP. Now during the electron transport chain, FADH2 is also oxidized. But it's oxidized at a different protein, a protein called Q. And it's located a little bit further down the electron transport chain. So that the electrons from FADH2 only pass through two protein pumps. Therefore, they cause two protons to get pumped, and ultimately they power the creation of two molecules of ATP for each molecule of FADH2. Once the electrons from either electron carrier make it to the final cytochrome in the ETC, they have to be removed. because if they weren't removed, that final cytochrome would get more and more negative and eventually would begin to repel other electrons that are trying to move toward it. So the process of removing them is important. And oxygen is the molecule that helps to remove those electrons from the final cytochrome. Remember, oxygen is really good at removing electrons from things because it's highly electronegative. Oxygen's role, the only role of oxygen in respiration, is to act. That's what we call the final electron acceptor. So the oxygen molecules will remove the electrons from the final cytochrome. They become negative, attached to some hydrogens to create water. So this is where the water made during cell respiration comes from. The electron transport chain can produce a maximum of about 34 ATP molecules for each glucose that initially entered glycolysis. And like we said before, it also creates some water, and it regenerates the NAD plus and the FAD that are needed to carry out other parts of cell respiration. An important term that you need to be familiar with is called oxidative phosphorylation. The electron transport chain and chemiosmosis together are sometimes referred to as oxidative phosphorylation. This term is very descriptive because during this process, the electron carriers, NADH and FADH2, are oxidized. And the energy from them is used to carry out the phosphorylation of ADP to make ATP. This is an important example of what we call energy coupling. So we've got the reaction of oxidation coupled with the reaction of phosphorylation. So here's a little look at how this process of electron transport chain happens. So here's a proton pump, another proton pump, and another proton pump. So NADH within the matrix is oxidized. The electrons move here, here, here. As they move, they're powering the pumping of hydrogen ions from the matrix out into the space between the two mitochondrial membranes, the inner membrane space, building up a high concentration of H pluses out here and a low concentration in here in the matrix. That happens again at the second pump and again at the third pump. And as I said a minute ago, FADH is oxidized a little bit further up the chain, right in here. So this would be protein Q. Its electrons only travel through this pump and this pump. Regardless, though, it still helps to build up this gradient, this H plus gradient. So since the H pluses can't go back in through this phospholipid part of the membrane, they shoot through ATP synthase. And as they shoot in, their kinetic energy is used by that enzyme to phosphorylate ADP to make lots and lots of ATP. That makes most of the ATP that our body uses. Finally, oxygen gas will go to this last cytochrome, remove those electrons, act as the final electron acceptor, and form water. And that's aerobic cell respiration. This sort of sums it up. So the theoretical yield of ATP for each glucose we start with, glycolysis nets us 2 ATPs. The Krebs cycle nets 2 more, and then the electron transport chain can net up to about 34, 32 per glucose. So in general, one glucose can yield up to about 36 ATP molecules. Now, even though we usually think about glucose as the original source of energy for the body, proteins and lipids can also be used as those initial energy sources. In order to use proteins for energy, the body must first break the proteins down into individual amino acids. The amino acids then undergo a process called deamination. During this process, the amino group from the amino acid is removed. That leaves behind a 2-carbon skeleton that is almost identical to an acetyl group. That 2-carbon group attaches to coenzyme A and then enters the Krebs cycle essentially as acetyl coenzyme A. And from that point forward, the process works essentially just like it does with glucose. The removed amino group is converted to ammonia, NH3, which is pretty toxic. And because of its toxic nature, the liver and the kidneys help our bodies to neutralize it and remove it. Lipids, on the other hand, can also be used. Lipids are composed of a glycerol molecule and these long chains of carbons and hydrogens called fatty acids. To use lipids for energy, the long carbon chains, the fatty acid chains, are essentially cut up into 2 carbon skeletons. This is called beta oxidation. These 2 carbon skeletons are attached to coenzyme A and essentially form acetyl coenzyme A, which can then enter the Krebs cycle. From that point forward, the process works essentially almost just like it did with glucose as the initial energy source. So here we can see that happening. This one's showing you the typical way we think of energy production. Here's proteins. So proteins are broken into amino acids. They're deaminated and used to create ATP. Here we see the beta oxidation process of fats. Notice that all 3 form an intermediate that's essentially an acetyl group, which will enter Krebs cycle, go through Krebs cycle, and then the products of that will enter electron transport chain. The last thing we want to discuss in this presentation is the details of anaerobic cell respiration. Remember, if no oxygen or not enough oxygen is present within the cell will carry out some anaerobic respiration. There are a few types of cells that solely use anaerobic respiration to make ATP. But this typically only works for very small and very inactive organisms. And the reason for that is that anaerobic respiration is not very efficient. It only yields about 2 ATPs for each glucose, whereas aerobic respiration is about 18 times more efficient. It yields about 36 ATPs per molecule. So larger and more active organisms still do anaerobic respiration sometimes, but they do it not as the sole ATP source, but rather as a supplement. They use it to supplement ATP production when oxygen levels are low. This might happen when an organism is running really, really fast or it's exerting its muscles at full force. The process of anaerobic respiration occurs in 2 phases. Those are glycolysis and fermentation. Glycolysis in anaerobic respiration is just like it is during aerobic respiration. The process breaks down the glucose, produces 2 net molecules of ATP through substrate level phosphorylation, 2 3-carbon molecules of pyruvate, and 2 molecules of NADH. When oxygen isn't present, the electron transport chain can't happen. And so the NADH produced during glycolysis can't be oxidized back to reform NAD+. Well, there's a limited amount of NAD+ in the cell. And if you run out of it, glycolysis stops. So there has to be an alternate method to regenerate NAD+ from NADH. That's the role of fermentation. Its only role is to free up NAD+ so that it's available to keep glycolysis going. The way it works is pyruvate serves as an electron acceptor and is used to oxidize NADH back to NAD+. Anaerobic respiration only yields the 2 net ATPs made during glycolysis. The fermentation step makes no ATP. There are many types of fermentation. We're going to talk about just 2 of them: alcoholic fermentation and lactic acid fermentation. During alcoholic fermentation, the pyruvates or pyruvic acids are used to oxidize NADH back into NAD+. We can see that happening here. So here's the pyruvic acid. It's being used to oxidize the NADH to make NAD+. In the meantime, the byproducts of this process are that the pyruvic acid forms some carbon dioxide, which bubbles out as a gas, and some ethanol, a 2-carbon alcohol. This is also known as drinking alcohol or grain alcohol. Yeast and some bacteria carry out this process. Beer and wine and some types of bread are produced using the products of alcoholic fermentation. Lactic acid fermentation, on the other hand, also uses the pyruvate molecules or pyruvate molecules to oxidize NADH back to NAD+. But instead of releasing carbon dioxide gas and ethanol, the waste product of lactic acid fermentation is an acid known as lactic acid or lactate. Most animals and some bacteria carry out this process. If an animal is using its muscles very heavily and oxygen levels drop, it starts to do this process, and the lactic acid starts to build up in the muscles, and it can burn. This is one of the reasons that we get very sore after strenuous exercise, and eventually we'll have to stop because of the pain caused by the buildup of this lactic acid molecule. The products of bacterial lactic acid fermentation have been used to create food products like yogurt, sour cream, buttermilk, and even some types of bread like sourdough bread. Remember that in animals, in large active organisms, this process doesn't produce enough ATP to be the sole source of ATP. It's supplemental, and it really only happens to a large extent when oxygen levels are very low. So this chart compares aerobic respiration and anaerobic respiration. In aerobic, oxygen is required as a reactant. It's not required in anaerobic respiration. In aerobic respiration, the substrate is fully oxidized. In anaerobic respiration, it's only partially oxidized. You're left behind with partially oxidized waste products like lactic acid or ethanol. The end products of aerobic respiration are carbon dioxide and water vapor or water. And the end products of anaerobic respiration are ethanol and CO2, or maybe lactic acid, depending on the type of respiration. In aerobic respiration, there needs to be an exchange of gases between the environment and the organism. In anaerobic respiration, not as much. In aerobic respiration, metabolic water is formed. It is not formed during anaerobic respiration. Aerobic respiration occurs partly in the cytoplasm, partly in the mitochondria. All of anaerobic respiration occurs in the cytoplasm. Aerobic respiration can yield up to 36 or 38 ATP molecules per glucose. Anaerobic respiration only yields the 2 that are netted during glycolysis. Aerobic respiration involves an ETC, an electron transport chain. Anaerobic does not. Aerobic respiration runs continuously throughout your life. It happens in plants and animals, all aerobic organisms. Anaerobic respiration occurs continuously in some really small inactive organisms, but it only takes place temporarily for short periods during oxygen deficiency in larger, more active organisms. That's our presentation on cellular respiration. Hope that was helpful.